Welcome back all. Let's continue with the cell signaling course and start from where we left. If you recollect, we were discussing different types of receptors, what are their activity association, and what exactly they do when signal comes in their contact. Their dimerization, heterodimerization, activation of signaling, generating second messengers, and so on. So when the signal and the receptors are in close proximity, they will do the signaling. How do you regulate it? Remember, we discussed that if the receptor continues to be phosphorylated, it will create a problem. For that, we need to do a dephosphorylation event. However, if we discuss further that before this event comes in, that ligand or signal binds to receptor, one can impose certain regulation, whether it is signal, whether it is receptor, or the effector molecules. So in today's class, we will see how the signaling is regulated, in which we will see that how different components of the signaling are regulated. So one of the simplest way you can think about of regulation is controlling the activity period of whether it is signal, whether it is receptor or effector molecule. While we understand that receptor should be more stable, but signal and effector molecules, their activity can be regulated. And one of the simple things which can be done is to have a control over the half-life of the receptor. So this receptor, one of the simplest things which we can do is to have a control over the half-life of either the signal or the effector molecule or signaling molecule. Like here, for example, the acetylcholine, which is one of the neurotransmitter we discussed in the previous class, that how it was affecting the muscle contraction in skeletal muscles, how it was affecting the heart and controlling the heartbeat, as well as how it was regulating the salivation process in the salivary gland. So acetylcholine, which is released in the synaptic cleft, consider the skeletal muscles, the half-life is 2 milliseconds. It's very small time if you consider as 2 milliseconds for this molecule to work, to bind to its receptor and activate it. Moreover, that the enzyme which is present over there so that it can degrade the acetylcholine has much stronger half-life. So you are putting together two things, one which is supposed to do the signal with a smaller half-life and one which is supposed to stop the signaling, the enzyme which will degrade the acetylcholine with a much larger half-life. So basically you are controlling the time which signal can spend with the receptor. So if we put that in perspective, generally this acetylcholine, which is synthesized inside the cell using acetyl-CoA and choline molecule with mitochondria helping this, and the enzyme choline acetyltransferase. This acetylcholine which is synthesized is packaged into synaptic vesicles. These vesicles fuse and release the acetylcholine in the synaptic cleft. Here the receptor for acetylcholine will bind to it and produce the signal inside the cell. Now you remember this acetylcholine esterase enzyme which is present in the synaptic cleft. How strong it is in terms of how long it is stable, and its activity, it hydrolyzes almost 4000 acetylcholine molecule per second. So this is the way you can actually control the signal right here, because the signal which is produced or generated is having a very small half-life. You can also consider that the availability of signal in terms of how much it is being produced, that is also a way to regulate the signaling. If there is less number of signal molecules, the signaling will be poor or it may not be at all present. But if you have to have signaling going on, you need so many more molecules above threshold so that signaling can go on further ahead. Another thing which helps in regulating the signaling, what we saw previously is half-life. Often that is more workable hypothesis for signals because they are transiently produced, they serve the purpose of binding to the ligand and activating the signaling, and then they are degraded. Similarly, the molecules which need to serve as a signal, or the ones which are signaling protein present inside the cell, or the effector molecules which are present inside the cell, they can be produced such that they are not active. Even though the amount present in the vicinity is in abundance, but they cannot initiate the process. If you think about the signal, one of the things signal should do is to bind the receptor. But if the protein is produced as a precursor, what we know for certain enzymes as a zymogen, so then although the signal is available, but it cannot bind and it cannot activate this. What we know about the zymogen or precursor like molecule, particularly in case of digestive enzymes, most of these enzymes are very active. They can cut around whatever molecules is available which may harm our gut. That is why most of them are produced in the form of zymogen. When there is a right set of environment available, they are activated so that they can start the digestion process. So like one of the most important molecule or the enzyme which is there is trypsin. This trypsin is produced as a inactive precursor or a zymogen which is called trypsinogen. This trypsinogen is activated by this enzyme enterokinase, and these enterokinase protein is produced by a set of cells which are called brush border cells. So the digestive system should receive the signal that trypsin must be activated so that protein digestion can be started. So the brush border cells will be activated. In that case, enterokinase will be released and trypsin will be generated from trypsinogen. Now what is the job of this trypsin? This is a protease basically. It can take protein and chop and make it smaller fragments. So what it finds that several of the enzymes which will help in the digestion of proteins, different classes of the enzyme, they are produced further as their zymogen or inactive precursor, whether it is pro-elastase, pro-carboxypeptidase or chymotrypsinogen. Trypsin works on all of them and removes this component which was making these molecules inactive. So chymotrypsinogen is converted into chymotrypsin, active starts digesting the molecule, pro-elastase is converted into elastase, start digesting the protein molecule. So basically controlling the signaling pathway by producing the signal or the effector molecules as a precursor is one of the ways to regulate the signaling. Now another thing which is often seen in the signaling world is that you can regulate the signaling by having an inhibitor, and this inhibitor could be for the signal, it could be for signaling molecules, it could be for effector molecules, it could be for kinases. You saw kinases are inactivated by dephosphorylation process, so you can have a dephosphatase enzyme over there which will inactivate the function of kinase. Also one thing you notice is that receptor dimerization was one of the things to activate it. So even dimerization could be served as a regulation for the receptor activation. But here if we think about the presence of inhibitor, so inhibitor will not allow the enzyme to function. We all understand the function of inhibitor for the metabolic enzymes or the enzymes which use a substrate and convert into the product. So one of these molecules we saw in the previous slide is the trypsin, which was being generated from the precursor trypsinogen, right? So once the trypsin is made, it is super active enzyme. What to do, how to control that? So the system has this anti-trypsin like molecule. What the name suggests is that the action of this antitrypsin is opposing the function of the trypsin. So basically you will have relatively more antitrypsin when this enzyme has to be inactivated, and once it inactivates trypsin, it will also affect the elastase, and we saw carboxypeptidase. So eventually bringing down the entire thing to a close. Similarly, this inhibitor which is or if I say that antitrypsin which is present in the gastrointestinal tract is basically serving as an inhibitor. We also know that there are gastric peptide inhibitor which actually affect the gastric juice formation. We all understand that how low is the pH in the digestive tract. If enzyme is active, always it will be a problem. Over there, pepsin is also produced as a precursor which is pepsinogen, and it is converted into the pepsin. So once this pepsin is produced, it is active, now it has to be shut down, so you need these gastric peptide inhibitor to do the job. So what you understand is that you can have a presence of inhibitor to ensure that the superactive molecule or the signaling which is started is culminated, or you can say that something which was supposed to be active, how to make it inactive. Now what basically this inhibitor is doing is this. We saw in the previous slide that zymogens are converted into the active molecule. They help these active enzymes, they help converting substrates into the product, and this inhibitor will ensure that enzyme is no more active. So how does signaling come into picture? You may think that it is all metabolic activity. What happens in case of signaling? One of the important molecules which we often see affecting this signaling or key effector molecule is called NF-kappa B. Normally NF-kappa B is bound to its inhibitor called I-kappa B. So when this molecule needs to be kept in inactive state and need not be signaling, it is bound to the NF-kappa B is bound to its inhibitor I-kappa B, and thus only when required the signal will come and remove this I-kappa B inhibitor so that NF-kappa B can be activated. Similarly, there are domains which not necessarily have an inhibitory function, but they are also called regulatory function. So these regulatory proteins, you can consider that I-kappa B to be a regulatory molecule, but it is regulating the activity of the NF-kappa B. Here protein kinase A, this particular molecule is a heterotetramer type of protein. So the two subunit of this heterotetrameric protein is the one which has the catalytic activity, and the two subunits of this enzyme is having the regulatory subunit. You will see the regulatory activity of this particular molecule when we understand a specific type of GPCR signaling later in the classes. So what I am suggesting here is that the presence of inhibitor or regulatory protein, they control the functioning of not only signal but effector molecules, and in this case you can also say that they may affect, presence of such regulatory proteins may affect the functioning of the receptors also. Then there is a thing which is called compartmentalization. What you understand from compartmentalization is that you need receptor and signal together to do the signaling. If you just segregate them, put them in different compartment, in some sense you can say that receptor is membrane bound. If you keep the signal in the cytosol, there will be no signaling, or if it is not available in the extracellular environment, there will be no signaling. So you have to put the two entities away. Only when the signaling is required, desired cues are available, they will be allowed to come in close proximity and do the signaling. So subcellular compartments are there which ensure that signal is not available. Like in case of neurotransmitter release, we know that neurotransmitter filled vesicles are present in the membrane vicinity, but they do not fuse and they do not release the content which is neurotransmitter. Only when their action potential arrives, that's the signal for these vesicles to fuse, and thus you will see that once the receptor is activated or binds to the desired molecule, this vesicle fusion will happen and neurotransmitter release will happen. Otherwise, this vesicle filled with neurotransmitter will be kept away from this active region where receptor for this neurotransmitter are waiting to do the job. Another thing which is there is that inside the cell there are calcium stores, the rough endoplasmic reticulum. Calcium is very important divalent cation which is used in many regulatory processes, including the enzyme activation and functioning as a cofactor at many places. So this calcium is kind of stored in this region which is rough endoplasmic reticulum. When there is a need for calcium, they will be released from this. For this, if you consider this as your cell, this is your plasma membrane and this is your RER, this is your nucleus. Now when there is a signal which binds to the receptor and activates a process, this process if requires calcium, it will generate things which will open these channels. If I say the channels for calcium, the calcium will be released and that calcium will be used locally. What this calcium can do, it can actually activate. One of the things is that it can activate the vesicle fusion to the plasma membrane. It can bind to certain proteins which are inhibitory or regulatory in nature and thus allow the activation of the target protein. Another thing which is seen with the mitochondria, that it hosts certain molecules in itself which are protected over there because they could be harmful to the cell. So if there is a signal for apoptosis or for death, these molecules will be thrown out of the mitochondria. We all understand that mitochondria in this case is a very unique organ which has two bilayers. So if I say bilayer 1, bilayer 2, there are two bilayers. So things inside are very tightly compartmentalized. Only when there is a desired signal, they will be coming out and mediating the process as desired. The other very important thing which I mentioned some time back in the form of phosphorylation, dephosphorylation, which comes under the broad topic of post-translational modification. This is one of the most common way to regulate whether it is signal, whether it is receptor or it is effector molecule or anything which is participating in the signaling process, right? So the proteins or peptides, nucleic acids or anything which is participating in signaling, whether it is a signal, whether it is a receptor, whether it is a effector protein, I am saying it again and again, they undergo this phosphorylation dephosphorylation cycle. I mentioned that the phosphorylation is attachment of the phosphate group. So what happens is that a set of enzymes which are called kinase, they utilize ATP, hydrolyze it to ADP and the phosphate group is released. This phosphate group will be put onto the target molecule. This target molecule could be an amino acid, it could be a protein, it could be a DNA, genetic material, or it could be sugar, it could be lipid molecules. So many different things undergo phosphorylation. So here using the ATP, the kinases use ATP. There is another group of enzymes called phosphorylase. They use inorganic phosphate. For transferring the phosphate group to the desired molecule. So you can consider this phosphorylation and dephosphorylation cycle as the most accessible transient kind of regulation on the function of components of the signaling process. So you can also correlate this with a process. I will come quickly to that. What this phosphorylation often does is that it may activate the protein which was inactive, or it may inactivate the protein which was active. So don't consider the phosphorylation as a process which will always activate the system. It can activate, it can deactivate also. Certain molecules are active when they are not phosphorylated. When time comes, they need to be phosphorylated and kept inactive. So I always give this example that the phosphorylation and dephosphorylation cycle is nothing but a game of statue, where phosphate is a statue statement. So once the statue statement is given, the person is inactive, does not move. And for coming back from the inactive state, somebody has to say relieve, right? The same thing happens here. The phosphate group added can actually make the protein move from one state to the other state. To bring it back to the other state, from inactive to active or active to inactive, you will need removal of this phosphate group. Again, I am emphasizing that phosphorylation does not say that the protein or the target molecule will be activated. It also equally is important in inactivating the target molecules. What all different things or the residues where phosphorylation can happen? So if you talk about the proteins, the amino acids are the target. You would realize that serine, threonine, tyrosine, these are very important amino acids which in bulk get phosphorylated. The most commonly phosphorylated residue is serine and threonine. Then in different organisms, you will see a good amount of tyrosine which is getting phosphorylated. In some situations, you notice that even histidine is phosphorylated, and what you can say is common between these three residues is the hydroxyl group. So that hydroxyl group is utilized for attachment of the phosphate residue. So when you talk about the protein, whether it is a proteinaceous signal, the receptor which is getting phosphorylated, or the simplified version of these signaling molecules, effector molecules, they will, if proteinaceous in nature, they will ensure that phosphorylation probably is happening these places to get the regulation going. Even the nucleotides which are phosphorylated by this enzyme, nucleoside diphosphate kinase, sugars, they are phosphorylated. We know the typical enzyme hexokinase which we have seen in case of glycolysis. Also the phosphoinositide moieties, they are phosphorylated, particularly phosphatidylinositol 3. So there is a class of enzyme which is called phosphatidylinositol 3 kinase. They will take lipid moieties like PIP and make it PIP 2, 3, 4 kind of thing. So you can generate a phosphorylated version of all biomolecules which are known, and from its unphosphorylated version. They could serve as a activating signal, they could serve as a inhibitory signal. So when the purpose is met, you need to dephosphorylate them, and that's where you will need the enzyme in the class of phosphatase. If you remember, I said some of the receptors also carry intrinsically the phosphatase activity. Other type of post-translational modification which is commonly seen is attachment of a methyl group, and that's called methylation. Similarly, you can attach an acetyl group and it's called acetylation. These two are very small entities which are being attached to the protein, very much like the phosphate group attachment. So a very small group which is getting attached over there. What is also understood very well is the post-translational modification by another protein. Rather than small moiety like methyl or acetyl or a phosphate group, there are entire protein which gets attached to another protein. Ubiquitin, we all understand that it is around 76 amino acid long polypeptide which gets attached to the target protein. You also understand, I am sure all of you know, that attachment of ubiquitin, particularly the polyubiquitin form, leads to the degradation of protein, right? So like compartmentalization was one of the way to regulate, even degradation is another way to regulate the signaling process. SUMO is nothing but a molecule which is very similar to ubiquitin, is called small ubiquitin like modifier. They also undergo attachment to the target protein as a monomer or a polymer fashion, and they have a function which is very different from ubiquitin, which is like we saw for polyubiquitination, the signal was degradation. Here often the molecule could lose its enzymatic activity, or it may induce new interactions, or the localization of protein could change. Remember the compartmentalization. If there is a cytosolic protein and SUMO gets attached, now this moves to the nucleus and compartmentalization happens. That's how the signal can be regulated further. Then you can have proteins which are attached to the glycosyl group or glycosylation. Basically you have sugar moieties or big carbohydrate groups which are getting attached to the protein residues, right? Protein different amino acid residues acquire the sugar and the carbohydrate moiety and then get regulated. In all these cases, what is happening, you are generating one form from an existing form. The other form which is being generated has small attachment or a big attachment, or maybe sometimes very big attachment and the polymeric form. This will end up regulating the function of that target molecule. What one has always appreciated is the presence of intrinsically the cysteine residues which undergo disulfide bond formation. So disulfide bond which is formed between the two cysteine residue is very critical, and insulin is one of the classical example where two chains of insulin are interconnected by this sulfahydryl linkage. So you can consider that the modification could be of a very small nature or a very big nature. In the previous class we saw that how signal could be very small like a photon, or it could be a big molecule. Similarly here, the modification of the signal or the receptor or any effector molecule which is regulating the signaling could be very small like sulfahydryl group coming together or a methyl group, or it could be a big protein altogether which is getting attached. So if you see where we have seen such modification regulating, histones are one of the most widely studied in this situation. The octameric structure of this histone is like you have H2A, H2B, then you have the linker H1. So the wrapping of the histone core which is there and the wrapping of the DNA, each of these histone they have a tail, and this tail is prone to get modified. There could be one type of attachment, if you can consider this as a methyl, there could be other thing which is acetyl group, or there could be something which is either attachment of ubiquitin or SUMO or something else. These are highly positively charged amino acid. The attachment of SUMO or ubiquitin happens on the lysine residues. So histones are positively charged, they have lot of lysine residues where ubiquitination or SUMO or SUMOylation can happen. If you look at a target molecule, a protein, it can undergo all these modifications whether by sugar attachment, acetyl group, methyl group, ubiquitination, SUMO attachment or disulfide group formation. So what you probably understand that there are many ways by which signal, receptor or effector protein can be modified to regulate the overall signaling process. So what we can conclude in this from this lecture is that the different components which you see in signaling, they can be regulated by different modes. One could be their modification by post-translational modification, attaching methyl, acetyl, phosphate groups and so on, or you can actually inhibit them by the presence of an inhibitor. The signals and receptors and these signaling proteins, they all can be modified, not just one of those, right? The half-life thing which I spoke about, the short half-life, that is more valid for the signal, but other modification, post-translational modification, happens to all of these components of the signaling process. Further, this modification has a tight control over the signaling process. You may activate the signaling by modification, or you may inhibit the signaling by the modification process. So in nutshell, we understand that how the signaling process is regulated at different steps in terms of whether it is signal which is being regulated, whether it is receptor being regulated, or the effector molecule being regulated. Thank you.